Hi everyone, I am Dr. Sheela, Assistant Professor, PG and Research Department of Microbiology, St.Joseph Arts and Science College, Cuddalore. Now I discuss about the topic structure of endospore. Bacterial endospore are special, tough, dormant and resistant spore producing by some gram-positive bacteria of Firmicute family during unfavorable condition. Bacterial endospore, it is highly resistant adverse environmental conditions. Endospore, as the name suggests, are seed-like formation producing within the bacteria. There are two types of spores, endospore and exospore. Endo means inside the bacterial vegetative cell. Exo means outside the bacterial vegetative cell. Endospore are developed within the vegetative cell. Bacillus and Clostridium is the most studied endospore forming bacteria. The spore formation occur within the nutrition, such as sources of carbon and nitrogen are depleted. Bacterial spore are highly resistant to heat, dehydration, radiation and some disinfectant agent. Next, who is discovered the bacterial endospore? The bacterial endospore were discovered by John Tyndall in 19th century. He discovered endospore as the heat-resistant spores of bacteria which survived even after 100 degrees Celsius. He also discovered a simple cost-effective process to kill bacterial endospores is called as Tyndallization. Next one is characteristics of endospore. The endospore are structurally, metabolically, functionally, it is very totally different from bacterial vegetative cell. The main characteristics of bacterial endospore are given below. Endospore are exceptionally resistant to stressful environmental conditions such as heat, ultraviolet radiation, gamma radiation, chemical disinfectant and desiccation. Most of the endospore are viable for many years, even for 10,000 years or more. Due to this long viability and their adaptation to stress condition, most of the endospore producing bacteria are notorious pathogen. Notorious means highly disease causing ability. That is called as pathogen. Wiping with alcohol and hydrogen peroxide or boiling at 100 degree Celsius do not destroy the bacterial endospore. Bacterial endospore can be visualized under light and electron microscope. Next one is structure of endospore. The shapes spherical, oval, elongated shapes may be narrow or border bludge than parent cell. The structure of endospore is very complex since they possess multi-layer covering. The outermost layer of the spore is called as exosporium. The exosporium is the spore coat composed of several layer of protein subunits. The exosporium is the spore coat composed of several layers of protein subunits. This one is structure of endospore. Outer layer of exosporium, spore coat, cortex, DNA, spore core, core wall. The thickness of spore coat is one of the reason for high resistance of endospore towards heat, radiation and some disinfectant agent. The inner to the spore coat is the cortex. It is very large space occupied as the half in the spore volume. The cortex is composed of peptidoglycan. The innermost layer of space is called as spore cell wall or core cell wall. The spore cell wall cover the central protoplast or core protoplast. The endospore core has a normal cell structure as the vegetative cell. The core contains a ribosome and a centrally placed nucleoid as a genetic material. The core protoplast is metabolically inactive. The core only contains about 10 to 20% of water of the normal vegetative cell. Next one is, why is bacterial endospore? It is highly resistant to temperature, radiation and a highly disinfectant agent. The endospore contain high amount of tepicolinic acid and calcium 2 plus ion. In some endospore, about 50% of total dry weight of spore is contributed tepicolinic acid. In this tepicolinic acid in bacterial endospore not occur in free state, rather it forms calcium 2 plus ion. For a long time it was believed that the high concentration of tepicolinic acid is providing the heat resistant to the endospore. The calcium 2 plus ion and tepicolinic acid can stabilize the genetic material of the endospore. The large amount of small acid soluble DNA binding protein are reported to occur in the core of endospore. This protein can bind to the DNA of endospore and can prevent the DNA from heat, radiation and chemical substances. The cortex of endospore can remove the water from the core asthmatically. The dehydration can provide the heat resistance in bacterial cells. The thick spore coat can also act an impermeable barrier against the chemical compound such as hydrogen peroxide. The spore coat can also restrict the entry of many hydrolyzing enzymes into the core. The bacterial endospore also contain a high amount of DNA repair enzymes. In this DNA repair enzymes can quickly heal all types of DNA lesions formed in the DNA when the spores are exposed into the harsh environmental condition. Next one is spore formation. The process of formation of endospore is called as sporulation. The formation of endospore is complex process and it's completed in 7 stages. What are the 7 stages? Stage 1, vegetative under unfavorable condition. Stage 2, asymmetric cell division. Stage 3, engulfment of bacterial vegetative cell. Stage 4, formation of cortex. Stage 5, formation of spore coat. Stage 6 and 7, maturation of spore and cell lysis. This one is stage 1, formation of axial filament. The genetic material of the bacterial cell is oriented in the exact center plane of the bacterial cell. Next one is stage 2, the formation of septa. The plasma membrane invagination grow into lumen of cell and forms a septum is called as spore septum. The formation of septum result in the separation of small portion of DNA from the rest of genetic material. Stage 3, engulfment of spore. The membrane of the spore, the mother cell continues to grow, completely engulf the new formed immature spore. Thus, within engulfment, the spore is now covered by the 2 plasma membrane and an inner membrane space. Stage 4, the formation of cortex. The cortex formation is started between the membrane space of the 2 membrane Large amount of calcium and tibiculinic acid has also accumulated in stage 4. Next one is stage 5, the formation of protein coat. The protein coat is laid down over the cortex of the newly formed spore. Stage 6, the spore maturation. The core became increasingly dehydrated. The cells became metabolically inactive. And then stage 7, enzymatic destruction of the sporogonium and release the endospore. Okay friends, thank you.